Hope everybody had a good weekend, and if you're a mom, you got celebrated, or you celebrated your mom, whatever's appropriate, or both, I suppose. Just a reminder, last Friday we didn't have time for our usual Friday lecture quiz in here, so you're going to get it today at the end of today's lecture period, and it'll have a population genetics problem, and it'll have some questions on the genomes chapter, which is the chapter that we started on Friday. So anyway, I'll end the lecture a few minutes early for that quiz. Today in laboratory, you'll get your regular Monday 8-point weekly quiz. Thank you. Your regularly Monday 8-point weekly quiz on the lab we did last week, which was plasmid isolation lab. And today's laboratory activity is electrophoresis, and there'll be an exit quiz for that as usual. So you guys get 3 quizzes in this class today. Tell your friends what an awful class this is. You get 3 quizzes in one day. Although I might have mentioned this in this Bio 11 class that I teach last semester. One class got 3 quizzes and a midterm on the same day, which it was kind of an unusual situation, but it did happen for one of my sections. Okay, well, let's recap a little bit of what we covered on genomes last time, and then head into new territory. Let's see now. One of the topics that the topic we started off this chapter on genomes was something called differential gene expression. And the differential gene expression means that cells can express certain genes within their genome and not express others. The cells in your body each have around 25,000 cells. Each cell has your entire genome. So the cells in your body each have all 25,000 genes, but they only express about 1/5 of those genes at any one time. In other words, the cells have most of the genes not being expressed, not turned on at any one time. And the major point where the cells do their differential gene expression is at the transcription level, making the mRNA. In other words, if a cell decides it doesn't want to express a certain gene, it just never makes a messenger RNA from that gene, which kind of makes sense. So we call that regulation at the transcriptional level. And so we started reviewing a little bit about how transcription works in eukaryotes. If this represents the first codon, the start codon of the gene, and there's the second codon and the third codon, and yada, 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 until we get to the stop codon, we can refer to this as the upstream region of the gene, the part of the gene that's upstream of the start codon. And that area is called the promoter region. And in laboratory, we learned there were some sequences there, like there's often a sequence called the ta-ta box, and often a sequence called the cat box, that some proteins called transcription factors have to bind to. And those transcription factors are specific for those promoter sequences, like there are transcription factors that bind to the ta-ta box and the cat box and all of the other sequences you find in the promoter. And the reason why they are there is those proteins, those transcription factors, are involved in attaching the RNA polymerase II enzyme, the one that makes the messenger RNA, to the template strand. The RNA polymerase II in eukaryotes just doesn't have a binding site for the template strand. It cannot attach itself. And that's different. In prokaryotes, the RNA polymerase does attach right to the template strand. So anyway, here come these transcription factors, and they stick on to those promoter sequences, and with their assistance, they can attach the RNA polymerase to the template strand, click, and once it's on there, it starts making the messenger RNA. So we covered that major concept in some previous lectures, but what I told you on Friday was this. If these were the only proteins involved in attaching RNA polymerase to the template strand, they're not very effective. In and of themselves, these transcription factors are necessary for attaching RNA polymerase II, but they're not sufficient. They have to be there, but in and of themselves, they're not enough to attach RNA polymerase II to the template strand. So we need some more proteins involved in that. And to see where those other proteins are and how they work to initiate transcription, we have to go upstream of the promoter, I should say far away from the promoter, to find some other sequences called control elements and some proteins called activator proteins that bind to those control elements. And how far away from the promoter? Well, it varies from one gene to another, but in some cases it might be 10,000 bases or 20,000 bases or 30,000 bases, kilobases and kilobases away from the promoter, way far away from the promoter. We find these control elements. So these are DNA sequences, usually way outside the promoter region, but that are involved in initiating transcription nevertheless. And these control element sequences are often found clustered together into groups called enhancers. So several control elements together form one enhancer. And there are proteins that bind to these control elements, or the enhancer if you want to think of it that way, called activator proteins. And these activator proteins help attach the RNA polymerase II to the template strand. And each activator protein has a specific binding site for one of the control elements, whatever the sequence is, GAA, GGA, GA. One of the activator proteins will bind to just that sequence. And if this sequence is TTC, AGT, another activator protein will bind to just that sequence. Okay, so here are the activator proteins. They bind onto their specific control elements, and then they are going to also help attach the RNA polymerase II to the template strand. Now, the mystery is that since those activator proteins are way far away from the promoter, where the RNA polymerase attaches to the template strand, how in the world do they help to manage to attach the RNA polymerase II? And the answer is that the DNA gets folded. There are proteins whose job it is to bend the DNA around like this. Here's one of these proteins that bends the DNA to bring these activator proteins on their control elements closer to the promoter so they can interact with the transcription factors, Gesundheit, and the RNA polymerase II. So here come those transcription factors. They all kind of glom together. Transcription factors attached to the promoter sequences. That, and then the activator proteins also get involved in the complex. They also get involved, and only when all of those are assembled, the activator proteins and the transcription factors into one big glob, one big complex, then and only then do you get attachment of the RNA polymerase II to the template strand. So I guess I'd say that any of these transcription factors, or even indeed any of the activator proteins by itself, is not enough to get the RNA polymerase II to transcribe. You need them all working together to get the thing to work. And what they found is when they look inside the cells of our body, in every cell they find the same transcription factors. Every cell has transcription factors that bind to cat boxes and ta-ta boxes. Those are always there. But which activator proteins are present does differ from one cell to another. And so it's really the presence or absence of these activator proteins which leads to differential gene expression. Why some cells express certain genes but not other genes. And so that's why I said here in this part of your handout, it's the control elements. What I should have said is the control elements and the activator proteins that bind to them that are really the true regulators of gene transcription. That's where you find the differences between the cells that lead to differential gene expression. And to illustrate that point, I gave you an example of differences in gene expression between some cells in your eye and some cells in the liver, and obviously very different genes we expect to be expressed in those two cell types. In the liver, there's a protein called the albumin protein. It's one of the proteins found in your blood, and the liver makes it. So in the liver, we expect expression of that liver-specific gene. This is a different gene. It's called the crystalline gene. It makes for a clear protein that forms part of the lens of the eye. And so this is expressed in your eye cells, but not in your liver cells. And so they have the same promoter sequences, cat boxes and ta-ta boxes, but where they differ is in their control elements way upstream of those promoters. You can see that they're based on the color coding, different clusters of control elements. Now, again, bear in mind that all cells have all your genes, and so the liver cell has the gene for albumin and crystalline, and the eye cell has the gene for albumin and crystalline, but the eye cells express this gene and not the albumin gene, and the liver cells express the albumin gene and not the eyeball crystalline gene. And it comes down to the activator proteins that the two cell types have. In your eyeball, in those cells, they have the activator proteins to bind to the control elements of the crystalline gene, but they don't have all the right activator proteins to bind to the control elements of the albumin gene, so it just simply doesn't get expressed, but the crystalline gene does. And in the liver cells, it's the opposite situation. In the liver cells express, they have the activator proteins to express the albumin gene, but they don't have all the activator proteins to express the crystalline gene, so they only express the albumin gene and not the eyeball gene. Sound good? All right. Now, in a way, this is kind of a chicken and egg paradox, because you may say, all right, now I kind of understand why I have differential gene expression between the cells up here in my eyeball and down in my liver, but that comes down to which activator proteins they're expressing. And so, how does the liver figure out to express these activator proteins and not some other ones for the crystalline gene, and how does the eyeball cells express the activator proteins for these ones and not that one, or just sort of push the expression of the problem of gene expression back one level to talking about how do they express the different activator proteins that you need. Well, the answer is that there is no solid answer yet. This is still an active area of research. They do know that cells become differentiated into certain types like blood cells and neuron cells and muscle cells very early in embryonic development. And so, initially we all start off as a single cell in a zygote that had all the, just one set of activator proteins to express certain genes. But as we developed into embryos, somewhere along the line, cells somehow got signals about which particular activator proteins they're supposed to be expressing, and so that turned them into nerve cells and blood cells and muscle cells at a very early stage. But once a cell has differentiated into a specific cell type, once it starts expressing certain activator proteins, it retains that same expression essentially all throughout your lifetime. Muscle cells stay muscle cells because they're expressing muscle cell activator proteins. So that goes a long way towards explaining cell type specific differential gene expression. There was another type of differential gene expression that we talked about. There was developmental gene expression, differential gene expression, where certain genes get expressed only at certain points in your lifetime. For instance, you might remember there are fetal and embryonic forms of the hemoglobin protein, the one that carries oxygen inside your red blood cells, and those versions of the hemoglobin are only expressed before you're born, and after you're born you start expressing the adult type of hemoglobin. And there's also environmental specific gene expression. An example of that was the expression of the digestive enzyme genes in your stomach and your intestines. That these cells don't make too many digestive enzymes when your stomach's empty, but when you eat food, then those cells start expressing a bunch of genes for digestive enzymes to help you digest the food. And it's amazingly specific the cells of your stomach and intestines even tailor the exact types of digestive enzymes that they produce to meet the type of nutrients you're getting in your food. If it's a carbohydrate-rich meal, you make more carbohydrate digesting enzymes. If it's a fatty meal, you make more fat digesting enzymes. Well, they think what's going on here for developmental and environmental specific gene expression is that some of these activator proteins can be allosterically regulated, that the cell can turn on or turn off the activator protein based on developmental signals and environmental signals. And to illustrate that concept, let's imagine a carbohydrate digesting enzyme. So inside, well, this is actually Rod Oka. He teaches at MPC. I don't know why I'm using him as an example, but anyway, inside his stomach are some enzymes that digest carbohydrates, like the sugars in the corn that he's eating. So how does him eating the starchy meal, the corn, lead to expressing those carbohydrate digesting enzymes? Well, imagine this yellowy thing here is one of the cells in his digestive system, and here is a gene for making a carbohydrate digesting enzyme like amylase enzyme. And imagine this is one of the activator proteins that